Hello, dear students. The next technique used for the determining the bacteriological quality of water is nothing but the membrane filter technique and nothing but the MFT. We can also say MFT, MFC, sorry, membrane filter, membrane filter count or membrane filter technique. We can say nothing but this method was developed by the Germany during Second World War for enumerating the coliform bacteria in water and has been accepted as a standard method for the uh, coliform organism. The membrane filter assembly, it is nothing but the constitute of a filtration unit made up of a glass or stainless steel and the suction pump. As we can see, this diagram is nothing but the we can also call this nothing but the membrane filter assembly. Here nothing but the flask. Here are one opening for the that first assembly is inserted into that particular flask, and here we have to add a suction pump so that the water sample which we have added to the funnel, we can suck from that particular filter so that the organisms which are present in that water sample can be trapped in that membrane filter. A filter disc which is used for the membrane filter technique, it is made up of the nitrocellulose which is placed in the filtration unit, that is nothing but that funnel. The pore size of the filter paper is less than bacterial cell size, so that the membrane filter are available with the different grades or porosity, which is ranging from 10 micrometer to 5 micrometer. So depending upon what type of water sample which we have to filter, we can use different types of filter of that nitrocellulose acetate filter. So the filters of pore size zero point 0.1 micrometer are generally used for the IMFT technique, which effectively retain all bacteria. In case of turbid samples, pre-filters are used to remove the particles. So in case of water sample, which is we have to analyze if it is turbid or it contains some sort of suspended material. As our filter pore size is very small, we can trap and choke that particular pore, so that depending upon that we have to use pre-filter so that the suspended or turbidity we can minimize, so that we can carry out further process. A specific volume of water is filtered through the sterilized assembly placed with a membrane filter disc. The filter disc is removed and aseptically placed on the surface of nutrient medium, which is already prepared in the petri dish. The growth medium used in this case is nothing but the modified endo agar or EMB agar, used in methylene blue agar. The plates are incubated for 37 degrees Celsius. After incubation, the colonies develop on the filter disc, whereas bacteria were entrapped during the filtration process. So, the bacteria during the filtration process which get trapped on the membrane filter, we have to place that particular disc on the nutrient media so that they can observe the nutrition from the nutrient media and show their growth pattern. The nutrient pass through the membrane and assimilated by the growing cells. The number of coliform colonies developed on the selective medium indicates their number per unit of volume of particular water, which is drawn through the filter. Dilution may be prepared for much turbid water sample. As we have already discussed, dilution technique in case of standard weight count, if the water sample is more turbid and as we already know that there is a number of microbial load is more, so dilution technique can be used. So in this diagram, we can also see this is nothing but the membrane filter technique. These are already aseptically or properly sterilized. Here, that funnel we have to add, that is nothing but the membrane filter. Normally, we use 0.5 micrometer of that membrane filter, which is aseptically placed in that particular place. We have to add a particular water sample which we have to analyze, and as the water sample is poured into that funnel, it is start pass through that particular membrane filter, and the bacteria which are present in water sample get trapped on that membrane filter. So sometimes we need a suction pump, so that due to the choking of that pores of membrane filter, the water is not passed through that filter. So by applying the suction, we can filter out that particular water samples. After the proper filtration of that water sample, we have to aseptically use or pick up the particular membrane filter and aseptically place over a nutrient media, which is we have already prepared sterile nutrient agar plate, and we have to place that filter on that petri plate, so that the media which is available in that plate get absorbed by the mechanisms, and they show their growth on the that particular plate. In the sterile membrane, filter is placed in holding apparatus. So these are nothing but the we have already discussed. And for this, we have used EMB agar plate. Normally, the characteristic growth pattern of fecal coliforms we have already discussed. This is nothing but the metallic stain type, black centered, small colonies can observe in this regard. So this is nothing but the basic mechanism behind membrane filter technique. So here we can discuss about the what are the different advantages of membrane filter technique. A large volume of water sample can be examined by means of membrane filter technique. Hence, it is possible to detect even few number of index organism, or we can see in case of coliform organism or pathogens in large number of volume. It permits testing of large number of water samples. Membrane can be transferred from one medium to another, so that it allows the selection and differentiation of microorganism, so that different selective and differential media we can use, and result can be obtained more rapidly with respect to that of the MPN, as it required 72 hours, and for the AMFT we only need 24 hours. After 24 hours, we can get a proper result of your MFT technique. Here we can discuss about what are the basic differences between MPN technique and MFT technique. So here is in case of MPN, it slower require at least 48 hours for the result. In case of MFT, it is nothing but the more rapid test. Quantitative result will be obtained after 48 hours of incubation, or you can say 24-hour need for the incubation. More labor intensive, less labor intensive. That means this test is carried out in 3 different stages, that is presumptive, confirmatory, and completed test. And for that, we have to only filter out the particular water samples, and we have to place on the nutrient agar media. And after 24 hours, we can observe results. So that it require more glassware. In case of MPN, it required less glasswares. Uh, for this, for each step, we required more culture medium. For that, we only need a single nutrient agar plate, a nutrient medium plate. Uh, Here, result obtained by the statistical approximation, MPN table we can use for that purpose, and the result obtained by direct observation, so that it is a high precision type of test. We can call that nothing but the MFT, applicable to all type of water, while it is not applicable for the turbid water, as the membrane filter is pore size is very small, and due to that the pores are getting clogged, so that the problem may be occur. Uh, Next part we'll discuss it next unit.